All right, welcome everyone to a new episode of the Roscoe's Wetsuit Neuro podcast. I am your host, Toby Passman. Roscoe's Wetsuit Neuro is an applied neuroscience company that currently offers premium brain health coaching to clients interested in peak cognitive performance globally, and will soon be offering targeted neuromodulation services, including QEEG brain mapping, EEG neurofeedback, brain photobiomodulation, and more to clients in the Miami-Fort Lauderdale area starting November of 2021. Go ahead and check out roscoeswetsuitneuro.com to learn more and to book a coaching session today. On the show with us today, we have a very special guest, Dr. Nick Jikomes. Nick is Leafly's Director of Science and Innovation and holds a PhD in neuroscience from Harvard University and a BS in genetics from the University of Wisconsin-Madison. He's the host of a popular science podcast, which you can listen to for free at nickjikomes.com. So Nick, really happy to have you on the show today.
Thanks for having me, Toby.
So tell me what originally sparked your whole interest in cannabis and hemp, what got you interested in studying it as a medicine?
I've been interested in cannabis for quite some time. I mean, just sort of going back to my own personal history with the plant. I think many people that have interacted with cannabis are often captivated by it in certain ways. It's a very diverse plant. When you look at different cannabis plants, there's clearly many different varieties. They look different, they smell different, and they can even feel different. And so there's been sort of a lot of mystery and intrigue historically around what that diversity is at the chemical level that gives rise to those that diverse array of aromas and potential psychoactive and medicinal effects this plant can have. Because if you're familiar with the biology at all, the cannabis plant contains many different compounds. The most famous one is THC, which is the principal psychoactive agent, which is responsible for the intoxicating effects. But there's actually, dozens and dozens of compounds, other cannabinoids, terpenes, and other classes of compounds that this plant produces. Different types of cannabis plants will produce them in different ratios. And there's a lot of interest right now, academically and in the private sector, to sort of map out that diversity and understand which combinations of these compounds might be most suitable for different things, just different types of effects, both in terms of your personal use or, applied uses either in the product development space for consumer packaged goods or in the research world for medical applications. So I've always just been very interested in that area of plant chemistry and understanding, what the actual, chemical logic of that is and how it maps to the types of effects it has in a mammalian brain.
Quick question for you. Are strain names bogus as far as when it comes to the effects of cannabis? Is it more so related to this, as you were kind of talking about, the combination and different ratios of the different cannabinoids and terpenes? Is there anything to, an actual strain? Or, because I've heard a lot of people kind of in this space talking about how a strain, or indica sativa doesn't really mean anything. What's your take on that?
Yeah, that's a good question. So I've actually been working for some time now on a research project that looks into those questions exactly. And so I've been doing a collaboration with some scientists at the University of Colorado, where we've taken a bunch of cannabis laboratory testing data from 6 different states across the US: Washington, Oregon, California, Alaska, Michigan, and Florida. And so these are the testing facilities within each state that are responsible for actually testing the products that go into dispensaries. And so they measure the cannabinoid and the terpene content of many of these samples. And so we have many tens of thousands of samples that have been tested for cannabinoids and terpenes. And what we did with these questions in mind is we first literally mapped out the chemical landscape of commercial cannabis in the United States. So we basically just show how the different cannabinoids and terpenes are spread out across the different chemotypes or chemical phenotypes of cannabis that we see. So we use different analytical techniques to define some of those chemotypes. And then we simply ask, how well do the commercial labels that are used match up to the actual chemical profiles that we see out there? And so we investigated that question at two different levels. So for those that don't know, in the cannabis industry, commercially, there's some very popular labels that are used that are purported to tell you different types of cannabis that are out there, meaning that one type will have tend to have one effect, one type will tend to have another effect. And those labels exist on two main levels that you'll hear about. So at the higher level, you have the basic three categories for cannabis flower and other cannabis products that you often see in a dispensary, and that's indica, hybrid, and sativa. So the claim, if you were to walk into a dispensary, you will almost certainly be told by a bud tender, the people that are basically responsible for getting you a product and recommending something to you. You'll be told very matter-of-factly that indica strains will make you relaxed or sedated, sativa strains will make you energized or creative, and hybrid strains, being a hybrid of the two, will be somewhere in the middle. It's a very simple and intuitive system. The big open question is it true at all? The reason this, even before I answer that, but the reason this nomenclature exists is really due to the legacy of prohibition and how legalization has rolled out in the U.S. Because cannabis has been around for so long. It's been in the United States since early 20th century. We've had decades and decades and decades of seeds coming in, growers in the illicit market illegally growing this, cultivating this, hybridizing different strains, and very creatively and systematically in many ways creating new cultivars. So if you talk to a botanist, they'll often, everyone has like their own language. If you talk to a botanist, they'll talk about cultivars. If you talk to a traditional cannabis grower, they'll talk about strains, which is sort of a term co-opted from microbiology, right? Different strains of a virus, different strains of a bacteria. They sort of co-opted that and started using it to describe the different cultivars, the different types of plants that they were breeding. And then, more recently, you'll have scientists like Ethan Russo and others talk about chemovars. So, thinking about it not in terms of the physical features of the plant, which is traditionally how people in cannabis think about strains. Does it have broad leaves or narrow leaves? Is it short or tall? Does it have a relatively fast or relatively slow growth cycle? But thinking about it in terms of its chemical phenotype or its chemotype. Is it THC dominant or CBD dominant? If it's THC dominant, what kind of terpene profile does it have? And so if you think about it, it must be true that if there are psychoactive or medicinal effects of different types of cannabis, if there truly are different types that tend to have different effects on average, that must mean that their chemistry is different, right? Because the effects are going to be caused by the chemistry. So if you just sort of think about it, very simple logical terms, if it's true that indicas are different from sativas in terms of their effects, then if you go look at all of the indica samples and you look at all of the sativa samples and you look at their chemistry, there must be some kind of difference there, and as is often true on the internet for these things, depending on where you look, you'll get contradictory answers. You can find websites that'll essentially say indicas are more sleepy because they have more THC. And then you'll have other people say, well, sativas are energizing because they have more THC. So, and there's very rarely any data attached to these claims. People that are slightly more sophisticated will tell you, again, very matter-of-factly, well, indica strains tend to have higher levels of the terpene myrcene, and that's why they're sedating and blah, blah, blah. But again, very little data actually attached to a lot of these claims. And so we wanted to assess whether it could be true in principle, whether or not different categories like indicas versus sativas or different strain names could actually have different effects. And that means, do they actually have, on average, a different chemical profile? And so we just looked at the data. And what that means is we took in and looked at tens and tens of thousands, many tens of thousands of samples across several different states in the US, as I mentioned. We looked at their cannabinoid profile and we looked at their terpene profile, and we mapped out that chemistry. And then we simply asked, how well does the indica sativa hybrid label attached to many of those samples match up with the chemistry? And then we did the same thing for strain names. So that work is about to be submitted, and a preprint will go up probably this month. So that will be like available to read on the internet as it's going through peer review. But just to give you a preview, the basic answer is when you map out the chemistry of commercial cannabis in terms of cannabinoid and terpene profiles, it pretty much does not line up in any clear way with indica sativa hybrid. In other words, if you close your eyes and you pick a random strain off a dispensary shelf, that's an indica, and you close your eyes and you pick another one off a shelf that's a sativa, they're no more likely to be different from each other than they are similar to each other. Another way of saying that is if you compare that random indica to a random sativa, they're really no more likely to be different than if you pick two random indicas or two random sativas. Now, what's sort of interesting about that is one way that could be true is if all the THC dominant cannabis commercially is pretty much the same, but it's not. So there actually is a chemical diversity out there. That diversity is simply not captured by the indica hybrid sativa labels as they're used in the commercial marketplace very well. So there are different terpene profiles. We used some analytical techniques to define, at least 3 clearly distinct chemotypes of THC dominant cannabis. In very simplified terms, relatively speaking, you would talk about strains or cultivars that are very high in terpene called myrcene and often pinene as well. So there's strains that tend to have relatively high myrcene and pinene. There's strains that
Before we go any further, could we just introduce kind of a definition for terpenes for the audience?
Yeah. So it's worth talking about cannabinoids and terpenes. They're the two most abundant and widely discussed classes of chemical compounds found in cannabis, although not the only ones. So starting with cannabinoids, because they're the most familiar and probably the most consequential for the effects, right? THC, CBD, these are the things that one will likely be familiar with. These compounds are cannabinoids, so they are plant cannabinoids. If they're in cannabis, they're coming from the plant, and they're cannabinoids because they belong to a class of compounds that is similar to endogenous compounds in our own body called endocannabinoids. And so these things tend to interact in various ways with our endocannabinoid system, which is simply a receptor system in the body, one of many. And I'm not going to go into too much detail here because we could spend a lot of time on that. But these are compounds that just have chemical properties such that they interact with the endocannabinoid system in some way. They're non-volatile. So when you smell cannabis, you're not smelling the cannabinoids. THC and CBD don't have the right chemical properties to float into the air and into your nose. But they are psychoactive. Both THC and CBD are psychoactive. Some of the other minor cannabinoids, which are very minor and very rare, as we showed in our paper in commercial cannabis, might also be psychoactive as well. They have a variety of effects, and there's a lot of stuff out there that you can go read on that. But that's one class of compounds. There's a different class of closely related compounds, so sort of like a sister class of compounds. So they share certain properties with the cannabinoids, but they are a separate class. And those are the terpenes or terpenoids. You'll hear both terms, and they're approximately interchangeable. And terpenes are actually, you could almost think of them as like a chemical language that plants use to communicate with the outside world. So these are among the most abundant biomolecules found in nature, especially in the plant world. More or less any plant that you've ever smelled in your life, what you're smelling there, at least in part, is terpenes. So they often have names like pinene. So the smell of pine needles comes largely from a terpene called pinene. There's different forms of pinene. The smell of lemons comes, at least in part, from another terpene called limonene. And there are literally, hundreds and hundreds of terpenes out there in the world. If you go walk through the woods, you're just walking through, an invisible cloud of terpenes. And if you smell a flower, you're really smelling the terpenes. The reason that they have all of these distinctive smells is that the plant is really, if you were to anthropomorphize the plant world, you might say that the plants are intending to release these compounds in order to say something to animals. That might be to humans, that might be to bugs, and bees and things. Oftentimes it's the things that might want to eat the plant. So a lot of these compounds serve a defensive function for the plant. They often have insecticidal or, some kind of aversive quality to them that sends a signal to insects, for example, that says, don't eat me, more or less. They might attract other things that will help to defend the plant from those other critters. But the way they do that is they basically travel through the air, or they come in contact with a bug or an animal somehow, and they have chemical properties such that they interface with receptors in that animal. And oftentimes that includes receptors in the nervous system. So, the plant is not really evolved to get humans high or attract humans, arguably. But what it has evolved to do is repel insects and things that might want to eat it. And as just a natural side effect of making these compounds for that reason, these compounds, many of them, bind to receptors in the nervous system. And so in the context of a mammal like a human, these things can have psychoactive or medicinal effects. They probably have modulatory effects on some of these other compounds. And the basic idea is these plants aren't making one thing, they're actually making a whole set of compounds. They usually make dozens and dozens of these things, like a whole bouquet of them. And the reason for that is that plants can't move. And so unlike animals, they can't run away from their problems. So they deal with them pharmaceutically, essentially. So they don't have an immune system like we do with cells that go around and eat bacteria. These things often have antibacterial or antifungal properties as well. Plants, a huge problem for plants and for cannabis growers is mildew, for example. So a lot of these terpenes, are helping the plant in that way, combat mildew or mold. But anyways, these plants often have a particular profile. So any two strains of cannabis, any two plants out in the world, flowers, trees, whatever, they tend to have a chemical signature that defines that species or that subspecies. And that signature is tied to that plant simply because that's the combination of compounds that's most suitable for that plant species or subspecies to, protect itself and just survive out in the world. And that's why you have a diversity of terpene profiles, for example, that you see in different types of cannabis. You can imagine cannabis out in the wild, if you're in the Hindu Kush region of the Middle East versus in, Central or South America versus in, Southeast Asia, you have a different habitat, you have different physical conditions that you're living in, and you've got different organisms that are trying to eat you or compete with you. And so the particular environment the plant finds itself in means that it's going to have a particular set of chemical compounds like cannabinoids and terpenes that will be best able to help protect that plant. And so that's where the diversity comes from. And then, of course, with human hybridization, these things are crossed with each other all the time to create new forms of diversity. And that gives rise to the diversity that we actually see in the commercial marketplace of consumer cannabis in the United States. And that is really the data that we were studying. So you take many, many samples from that consumer marketplace of all the cannabis flower across the US, and we look at what is the set of all cannabinoids and terpenes that we see in these samples. And when we do that, as I said, it doesn't line up very well with the indica sativa hybrid labeling system. And so then the second question is, well, what about these strain names? Well, there's, allegedly thousands of strains out there. In other words, allegedly there are thousands of distinct types of cannabis. And if that's true, then there should be a bunch of clearly discernible chemical profiles or chemotypes, chemical phenotypes that we see. And if the strain names mean anything, if they are a reliable marker for the effects that a given strain will cause, then we ought to see that these strain names reliably line up with distinct profiles. So, for example, there's a very popular strain called Blue Dream There's another very popular strain called sour diesel. Sour diesel is commonly, I believe it's considered a sativa normally, and so people will often describe it as having energizing effects. Blue Dream is usually considered a hybrid or a sativa dominant hybrid, depending on who you asked. Maybe its effects aren't quite so energizing. And then you've got something like Granddaddy Purple, which is considered to be a quintessential indica that will have sedative effects. Well, if these things were all true statements, then when we go look at the chemistry of samples attached to those names, we ought to see two things. One, that all of the Blue Dream samples should be relatively similar to the other Blue Dream samples, right? They're not just haphazardly using these names. And the other thing is they should have distinct profiles between the strain names, right? So if we look at all the Granddaddy Purple samples, they ought to have some difference in their chemical profile compared to all the Blue Dream samples and all the sour diesel samples. And so we just took this very large data set, the largest data set, I think, of its kind that's been studied like this. And we looked at the set of all samples that had a strain name attached to them, and that was a very large set. And we said, how consistent are these strain names? Are people just randomly picking strain names and there's no systematicity to it at all? And how distinct are these different strain names when you look at them? So the basic answer is this. When you look at strain names, what you find is that they are a more consistent indicator of chemical profiles than the indica sativa hybrid labels, but they're certainly not perfect. And there is a huge spread among the strain names. So some strain names are relatively consistent, meaning if you close your eyes and you pick two different samples from two different producers with that strain name, odds are good that they will have the same basic profile. But there's other strain names where a much weaker relationship exists, and there's some strain names where you do no better than chance. And we can actually model this quite well, because what we can do with this data is we can just randomly scramble all of the strain names. We can randomly shuffle them across all of the different product IDs that we have and ask whether or not the actual strain names attached to the data are more or less consistent than that. And the basic result is it depends on the strain name. Some strain names are statistically more reliable in terms of pointing to a particular chemical phenotype than you would expect from randomly shuffling all the names or randomly just naming all the products. And some strains are not. And there's a huge spread there. So, just a huge distribution. Some strains are relatively reliable, some are sort of medium, some are low, and some are no better than chance. Many of them are no better than chance. So, there's a lot more we can unpack there, but what else? Do you have any questions so far?
I do have a couple. So as far as, when it comes to like your example of, sour diesel, generally people think of it as more like an energizing sativa, Blue Dream somewhere in the middle of as a hybrid, and like Granddaddy Purple, what you're mentioning is like a kind of more of a sedating indica. So it sounds like, the placebo effect is definitely coming into play, a big factor here as far as like, do you believe that, say someone who's going to, someone who's using commercial cannabis and they're going to a dispensary, so they've both probably heard of these different strains. Maybe they've looked them up on Leafly and read, this one's going to be really sedating, this one's going to be really energizing, and then they also go and the bud tender is telling them this is going to be really, that.
Yeah.
So it seems to me as if there's almost maybe like a twofold sort of a placebo effect going on, where people are kind of being led to believe that the strain is going to produce a certain effect, even though its chemical makeup might not be what's actually responsible for that effect.
Yeah, there's definitely very large expectancy effects, and that's not surprising. I mean, you would expect that going into this, right? This is an inherently subjective thing. I mean, when you're talking about a THC dominant strain, which is what we're talking about 95+% of the time, right? It's making you high. It's literally altering your conscious subjective experience. It's very difficult. I mean, we can all appreciate how difficult it is to talk about our subjective experience. And so it's inherently something that's very difficult to talk about and something that's very amenable to suggestion. So yeah, there's definitely a lot of expectancy effects, and that's not surprising in any way. But there's also, chemistry is really only one factor. Chemistry is an important factor in determining effects. Context and expectations are another important factor. But the other one that's big, and arguably even the biggest one, is dose. So, if you take two different cannabis products that are high in THC and they have a different terpene profile, say, and let's just assume, for the sake of argument, they will cause different effects. Well, if they cause different effects, that means that they will cause different effects for you at the same dose. But if you simply smoke enough of anything with THC, that changes the effects, right? A low dose and a high dose of something have different effects, even if it's the same thing that you're smoking each time. So, to put it in simple terms, if you smoke enough of anything, you're probably going to go to sleep. So then imagine someone, going into a dispensary. They're like, okay, I'm looking for cannabis to help me sleep. I want to get sleepy. They go in, the bartender says, use this one. It's an indica. It will make you sleepy. Well, whether or not it is that or is something else, if you just go home and you smoke a little bit and it doesn't make you sleepy, and then you just keep smoking more, you'll probably get sleepy at some point. So you can sort of tune, you can, in some sense, get many different effects out of the same thing by just modulating the dose. And, so that's just another complicating factor here. So, it's actually not difficult for someone to be like, oh yeah, that thing caused the effect I wanted, because they're expecting it to cause that effect. And, whether or not it's the best thing to cause that effect, you can probably get that effect to some extent at some dose for that strain. So there's all of these complicating factors here. And then all of this is riding on top of the fact that, these names are, there's really no stringency. So there's no, you can name a product anything you want. What's kind of remarkable about the results that we found is that there was any consistency at all to the strain names. The strain names aren't distributed randomly, and we can look at that directly in our data set and analyze it directly. So it means that people are actually, with some reliability, naming these things, in a somewhat disciplined manner. So, for example, let me give you some examples. A strain like Blue Dream is actually, what you describe as relatively consistent. So when you take, and we've got many Blue Dream samples, and again, when a sample is labeled Blue Dream in the data set, that doesn't mean it's bona fide Blue Dream. There's no genetic verification that cultivar is Blue Dream. The growers can name anything blue. They can put a tulip in a box and call it Blue Dream, and all we would see is the terpene profile for a tulip and the label Blue Dream. So there's nothing guaranteeing that these things really are Blue Dream. All we know is we've got a terpene profile and a cannabinoid profile, and someone said it was Blue Dream. Now, turns out that even though anyone can name these things anything, all of the many hundreds of Blue Dream samples we have, I don't have the exact numbers in front of me, but around 3/4 of the time, 75% or so of those Blue Dream samples are the same basic profile, even though they're coming from different growers in different states across the country. Blue Dream is most commonly associated with this high myrcene, high pinene chemotype. So it's THC dominant, and it tends to have myrcene and pinene as its predominant terpenes. And, remarkably, about 3/4 of the time, give or take, the Blue Dream samples have that basic profile. And it's not always the same, right? It's a little bit different, but that's that same basic profile as opposed to a distinct terpene profile. So the way you might think about this, if this is all unfamiliar terminology to you, is if you think about something like wine, and you think about the different varietals of wine, right? You've got Merlots, and you've got Pinot Grigios, and you've got Chardonnays, and so on and so forth. But there isn't just one type of Merlot, right? There's probably thousands of brands out there that have their own Merlot. And the Merlots aren't all identical to each other. They might even change for the same vineyard one year to the next. But we know what a Merlot is, and there's a definition of what that means. And, they're all relatively similar to each other, right? They all taste and look and smell much more similar to the other Merlots than they do to the Pinot Grigio, even though all the Merlots are not identical to each other. And that's what you see, at least with some of the strain names, that majority of them will be in the same basic chemical neighborhood, right? So you can think of them as like, okay, this one's usually a Merlot. It might be a little bit different from one Merlot to the next, but it's the same basic chemical phenotype each time. And then of course some strain names are not like that. Some of them are truly random. But what's interesting is, a strain like Blue Dream has a relatively consistently distinct profile from something like Super Lemon Haze, say. So Super Lemon Haze is also THC dominant, but it actually tends to have high levels of this terpene called terpinolene, and it has relatively low levels of myrcene and pinene, which were high in Blue Dream. So there are strain names out there where a majority of the time those samples will have distinct terpene profiles from samples with another strain name, but it's never perfect. And the only way to know for sure is to have that lab testing data.
Nick, let me ask you this. If you could sort of play God in the sense of kind of recreating the whole commercial cannabis space and strain names and labels for different products, do you have any ideas as far as how you think would be an ideal system? Like, would you
Mm-hmm.
abolish
Mm-hmm.
strain names altogether? Would you
Yeah.
reclassify them somehow? What's your take?
Yeah. So there's a wide range of opinions on this depending on who you ask. So, if you talk to someone, in the growing community, that will tend to be someone who, there's an entire culture that's decades and decades old of growing and hybridizing these plants, and doing that at great personal risk, because until very recently that was illegal. So there's a lot of time and a lot of effort that was put in by people who took on a lot of personal risk to create these cultivars that are circulating legally in many places today. And those are the people in the community that did the work to create these things and to name these things. And so they would say that the strain names are fun, they're part of the culture, and they do, or at least can, mean something. Someone who's more of like a hard-nosed, science or academic type, likes to scoff at the strain names and say, "Ha ha ha." Botanists don't use the term strains. That's what we call bacteria as having different strains. That's not the official way of talking about it. But language is malleable. And categorization is different. What we need is, we need, these are plants, right? So it's good to have that rigorous, botanical scientific perspective, but these are also consumer products that are being used by regular people out in the world. So, you might, someone who's more of a hard-nosed scientist might say, like, we should call them Chemovar 4.3A and Chemovar 2.6D, and like give them names like that. But that's not functionally useful to a consumer, right? No one, even if you tried to do that, the people talking about this out in the real world and buying these products and using them simply aren't going to adopt a system like that. It needs to be simple enough and relatable enough that the average person can use it, but it needs to be rooted in the science, and it needs to be rooted in something objective and real. And there needs to be some kind of enforcement of some kind of standard that's rooted in that science. And this is not an alien thing. Like, this exists for other things that we all buy all the time. Like, something like wine or apples or corn. I'm not an expert in those areas, but my understanding is, right, there are rules and regulations. If I'm, if I've got some apple trees outside and I want to start selling apples, I'm not allowed to just slap a golden delicious sticker on it. Like, it actually has to conform to some standard based on, what that seed is and what its properties are. If I'm selling wine bottles from my wine vineyard, I can't just arbitrarily call something a Merlot or Pinot Grigio. It's got to be made of the right grapes and have the right stuff in it. And I'm not sure exactly what those details are, but there's precedent for these things. Like, this has been done before. And so I think the right general approach is, you want to, the first stake in the ground needs to be genetics, right? So the genetics of a particular cultivar will allow you to confirm whether it is that cultivar, right? So if something truly is Blue Dream, it will have a genetic signature that tells you it's that cultivar as opposed to something else. So if you're growing something else, you should not be allowed to call it Blue Dream. But today you are allowed to do that, right? I can just pick a random cannabis seed, grow it, slap a blue dream sticker on the box. So that should not be allowed. But then you need another layer because two different blue dream seeds grown by two different people might not have an identical chemical profile, just because the grow conditions can modulate the profile to some extent. So you ultimately want the categorization to be based on the chemistry, because that's really what matters in terms of the sensory and psychoactive effects. And so my belief is that the chemistry should be front and center in defining what type of thing the product is labeled as. So we need some kind of system that's rooted in the chemistry first and foremost, but also ties that to the genetics of the cultivar. We need that system to be standardized across states and enforced by some body that's got the authority to enforce it. And we need to have that strong rooting in science, but we also need to be using names that, like, your average bud tender and your average consumer will relate to and be able to use in their own linguistic environment. So my general view is that even though strain isn't the official botanical nomenclature that a scientist would use, strain names are what's historically been used in cannabis. It's what's widely used today. And even though they're not used with total discipline today, and there's no real standardization that's been enforced, I think it's easy to imagine, relatively easy to imagine, that one could enforce such a standardization. And in other words, one could use at least some of the existing strain names, use them in a more disciplined way so that they do reliably, for all of them, refer to a particular chemotype each time. And just create the right regulatory guardrails to make sure that things stay that way, right? And that basically simply means that if you're going to grow something and call it something, you've got to know the genotype so that you know what cultivar you're growing. And then you've got to use names and labels, whatever those may be, we'll just be agnostic for now, in a way that consistently refers to a particular chemical profile. And that's not unlike, again, what you do with wine today. Like if I go buy a Merlot somewhere, it doesn't matter where I buy it, doesn't matter what brand it is, I know it's not going to look and taste like a Pinot Grigio. It's going to look and taste pretty much like every Merlot I've had. Not that they're all exactly identical. It's a general group, but there are guardrails that define what that means and ties it to objective features of the product I'm actually buying.
Nick, switching gears a little bit, I wanted to ask you, your take as a neuroscientist on just kind of uh, cannabis and the brain, in terms of there's, plenty of research I've read as far as the different cannabinoids have, having neuroprotective properties, along with some beneficial effects of a lot of the different terpenes. But then on the contrary, there's, studies about cannabis and short-term memory and, some, I guess, more detrimental effects of cannabis on the brain. And I just wanted to kind of hear, your take on, uh, is it good for the brain? Is it bad for the brain? Is it somewhere in the middle? Um,
Yeah. Well, I sort of reject the question. I'll answer a slightly different question. So, I don't believe you should be thinking about things as anything as like good for you or bad for you. So whether or not something is good for you or bad for you is highly context dependent. In the case of drugs and psychoactives, it's highly dose dependent as well. So, one compound can be very good for you at one dose in most contexts, and it can be very bad for you in a different context or at a different dose. And this is very true for some of these compounds. So, I think you simply should not think about individual drugs as being good for you or bad for you. The answer is pretty much any drug can be either, and it depends on how you're taking it, how much of it you're taking, and what the surrounding context is. So let's unpack that let's just sort of talk about two compounds concretely. Let's talk about THC because it's the most common one. That's in cannabis, and then maybe we'll pick a terpene to talk about. So THC, obviously, it's the principal psychoactive agent in cannabis. It binds to the CB1 receptor and it makes you high. It also does a number of other things because it's not just binding to the CB1 receptor, it's binding to a number of things in your brain. And the extent to which a drug is going to bind to all of the things that it binds to is going to depend on the dose, right? So if you imagine, say, 3 receptors that a given drug like THC binds to, maybe one receptor is the most sticky for that drug, and so it'll tend to tickle that receptor the most. But then if you keep taking more and more, you can saturate that receptor, and now you'll start to kind of activate other receptor systems. And that's sort of a crude way to think about why you get biphasic or dose dependent effects with something like THC, which you do. Meaning, if you take THC at a relatively low dose and a relatively high dose, you can get very different, even opposite effects. Right. And so that's one reason why I don't think it's good to think about drugs uniformly as good or bad, because the exact same drug can, in some cases, do more or less the opposite of what it did at a low dose if you take it at a high dose. So with THC, as a general rule of thumb, if you take it at a lower dose, the type of psychoactive effect you would people often experience at a lower dose is one that's more euphoric or maybe stimulating. It's not going to be overwhelming. As that dose, and it can actually be anxiolytic or anti-anxiety. As that dose goes up to a relatively high dose, it can start to flip in the opposite direction. It can become dysphoric and unpleasant. It can become anxiogenic. It can make you more anxious and paranoid, as many people know. And it can become sedative as opposed to energizing. So that's connecting that back to our earlier discussion. Why do some people say that some types of cannabis are energizing and some people say that some types are sedating? Well, a lot of that I think is explained by the dose of THC that you take and your own sensitivity. I think almost any THC product can be energizing or sedating depending on how much you take. And the level that you need to take for an energizing or sedating effect is going to depend on your own sensitivity, your own intrinsic sensitivity, and your own tolerance level based on your consumption habits. So, I simply don't think it's valid to say that like THC is good for your brain or bad for your brain. It does some good things, and many of these compounds do. So the cannabinoids, including THC, generally have very good anti-inflammatory effects. So THC, at least by some measures, is a more potent anti-inflammatory than many things that might be in your medicine cabinet right now, like hydrocortisone or aspirin. So a lot of these compounds have anti-inflammatory effects. That's why that they can help with things like different forms of pain. One of the most robust medical things that cannabinoids like THC help with is pain, especially chronic pain. They can disrupt memory acutely. So if we talk about memory and whether or not it's good or bad for memory, there's two things that come to mind here. One is acute versus chronic effects, and one is age-dependent effects. So let's take those one at a time. The acute effects of THC intoxication are pretty straightforward in terms of memory and cognition. It disrupts your cognition. Now, on the one hand, you can become more creative and you're in different thought patterns, and people, including myself, often have used cannabis, just to get the ideas flowing and the creative juices going. So there is value, in my opinion, for doing creative or intellectual work and just stirring the pot and putting yourself into a different mind space than you're normally in. But it's going to, while you're high, your short-term memory will be impaired. Your ability to sustain attention on one focused task will be impaired. So it comes with cognitive impairment in that sense. Now, again, that doesn't necessarily mean it's a bad thing. In the right context, it's okay to do those things, and we all do those things all the time, right? Like a little kid spinning around in a chair is impairing their cognition essentially, because they're not going to be able to walk around for a few seconds in a state of dizziness, but it's still okay to do that. Celebrating a wedding by getting drunk is something we've all probably done. In the right context, that's okay. If you're at home or in a safe context and you're doing something that impairs your cognition but allows you to appreciate sights and smells in a new way, or makes you feel more creative, or just makes you laugh, like that's all good. That's all fine. So again, right, there's like, do you talk about that temporary acute memory impairment as a bad thing? Oh, yeah, I mean, you can say that it's impairing your memory, but on the flip side, right, you can get a lot of benefit from it as well in the moment. But it's good to distinguish the acute effects that we describe from chronic effects. My understanding, this literature is always somewhat controversial, and different people will see different things in different studies. the chronic effects of THC, my understanding is that historically there have been studies that show that chronic long-term use of a lot of THC can cause chronic cognitive impairment, meaning, people are essentially scoring lower on memory tests and things. But my understanding is also that a lot of those effects seem to disappear when you control for other confounding factors. So I would say it's not completely settled science what the long-term effects are and whether or not there's any clear negative impact on brain structure and function in the long term. I would say that statement is probably limited to people using it as adults. There's generally, for THC or for any other drug, essentially as a good rule of thumb, if you start very early, especially adolescence or pre-adolescence, and you're using the drug chronically from that young age before your brain is, developed and matured, the odds of long-term detrimental effects are higher, and the odds of developing dependency is higher. That being said, right, if you're using it as an adult, it has many uses. So we touch on the fact that it's a good anti-inflammatory. It can also just generally help with pain through its effects on euphoria, causing euphoria and things like that. And then what's super interesting is these age-dependent effects we haven't talked about yet. So they've done some very interesting experiments in mice that I'll describe with THC, and they're doing clinical trials in humans now. But what they do is essentially this. You take young mice, they're basically what you would call adolescent mice, right? Young, healthy, spry. They do really well on learning and memory tests, right? They're sort of in the prime of their life. And then you take old mice. And we're going to do same sets of experiments in the juvenile or adolescent mice and in the old mice. And of course, the old mice, a little bit slower to complete tasks, right? They're not as they're not quite as good at the learning and memory tasks, and they're older. So what happens is they did this experiment that looked like this. They gave these mice THC chronically, so basically quite a bit of THC every day for a month. And then they let them sit for a week. So when they do the actual test, there's no THC in the system. These mice aren't high when they're doing this. They're in a sober state of mind in all conditions. So give juvenile mice THC for a month, and then a week later, when they're sober, you put them through a battery of learning and memory tests, right? You make them navigate through mazes and things like this. And what you find is that the mice that were given chronic THC when they're juveniles do worse than their peers who had not been given THC, that were given placebo. So in other words, chronic THC administration in juvenile mouse will tend to disrupt learning and memory. And those disruptions are measurable even in the absence of THC in the bloodstream. And then you see that in the brain too. So when they go in and they look at the neurons in the brain, they see cellular signatures of those learning and memory impairments, right? There's basically fewer connections between neurons, and the neurons just don't quite look as good, so to speak. That's like a classic, that's not a surprising result at all. That's sort of like the classic chronic THC administration result, that you tend to get acute impairment when basically animals are asked to do things when they're high, just like you might imagine you would get in a human. But chronic administration will also cause impairments in the animals that received it chronically compared to those that didn't. What's super interesting about this experiment is what happened in the old mice. So the old mice, essentially the exact opposite happened. The old mice when they're sober, tend to perform less well than juvenile mice. In fact, old sober mice sort of do as well as high juvenile mice, if I remember correctly. But the old mice that were given chronic THC for 30 days actually did better than their sober counterparts. And likewise, they saw indications of increased plasticity and things in the brain. So in other words, chronic THC administration to an old mouse had a kind of cognitive enhancing effect. And so the idea here is that if you administer THC at the right dose, you can actually get different, even opposite effects at a young versus an old age. And the sort of theoretical or the hypothesis behind that is, as we developed and as we age, our endocannabinoid system changes. The levels of CB1 receptors and things in the brain change. And that change in CB1 receptor levels among other things that probably change, is going to impact what and how THC actually does in the brain, such that, perhaps something that's detrimental at an early age can actually be beneficial at a later age. And I do believe they're doing clinical trials in humans right now, in older human patients, to see if low dose THC can actually have some cognitive enhancement effects. So basically the opposite of what you would expect it to do in a young person.
That's super interesting. And I would assume probably like neurodegenerative conditions that probably would have a lot of implications for that.
Mm-hmm. Yeah. And I mean, that's exactly right. So, um, it's sort of ironic that after all of these years, it could turn out to be true if these clinical trials match the animal studies that, THC, at least at a low dose, when you're elderly, might be quite neuroprotective. Um, I think it's plausible it will turn out that way, just because we know that these compounds can have certain anti-inflammatory and neuroprotective effects. Some of them act as relatively good antioxidants. It's not difficult to imagine ways that they might induce plasticity, or be stimulating in some way to neurons in the right aged individual. And again, it just comes back to this notion of, you can't just uniformly think about a drug as good or bad because of all of these factors that come into play. The dose is super important. It can make something good bad, or make something bad good, depending on the dose you take. And even if it's the same thing at the same dose, same everything, but you just wait 20 years, your brain might respond very differently than it did 20 years ago. So that's why I don't like to say that any of these things are good or bad. It's all very context dependent. And then of course there's other compounds. CBD is another one. That one is one where, like, there's very little negative effects you can find with CBD. Even when they give it to young children in these epilepsy trials, and they're giving them hundreds or thousands of milligrams, it doesn't seem to have any real negative side effects, nothing toxic. The worst you can say probably is that it can inhibit certain metabolic enzymes in your liver that influence the metabolism of other drugs. So you just need to be mindful of that. But we're talking about very high doses of CBD. So CBD, at the doses that the average person would be taking from a CBD product they might have, there's not much you seem to be needing to worry about. There's no evidence of toxicity. The only thing you really need to worry about is whether or not you actually have taken enough to do anything. But on the flip side, we can talk about some of the terpenes. So some of the terpenes are present at quite low levels in cannabis, and many of them have interesting properties. Many of them, beta caryophyllene, for example, can be fairly potent anti-inflammatory. It can interact with the CB2 receptor in the endocannabinoid system. But some of these compounds, limonene, some of the other terpenes, can be irritating and caustic at a relatively high dose. So if some people seem to take the viewpoint that, plant medicine is all good all the time, and, just give me more of everything. But, some of these terpenes will have, more or less corrosive effects, at least at a high dose. So, and some of them, I think, at the right dose can have shown, at least in vitro, evidence of some cellular toxicity effects. And after all, right, the plant is making these things essentially for the purpose of irritating bugs and things trying to eat it. So it's not surprising that some of these things are going to irritate our own cells as well. But again, a lot of that is dose dependent. And I strongly suspect a lot of these terpenes might have potentially beneficial effects at one dose, but then if you go too high, the potential irritation or harm starts to outweigh that benefit.
Nick, I want to ask you about as far as the interaction between, THC and CBD. So
Mm-hmm.
just as far as my, personal experience with, cannabis, adding a little bit of CBD, to THC, completely changes the effects for me and results, in a um, different sort of high. Can you tell me what's going on in the brain there?
Yeah. So there's fairly reproducible evidence in the literature that if you co-administer THC and CBD, you give them at the same time, or you give CBD before the ingestion of THC, that the presence of that CBD will impact the effects that THC has. So there's definitely like a modulatory effect there, and there's a kind of interaction that's happening. And we actually have fairly good knowledge of what the basis for that is pharmacologically. So THC and CBD are very different in isolation, right? THC is the cannabinoid that gets you high. That's sort of the classic cannabis cannabinoid that you think about. CBD is non-intoxicating. It is psychoactive because we know that it can affect mood and anxiety and things like this. So it's psychoactive but non-intoxicating, and it doesn't get you high. So it's sort of radically different from THC in some sense. People often talk about THC and CBD in scientists in the literature as having sort of opposite effects in many ways. The locus of interaction for them is probably at the CB1 receptor. It's not necessarily only there, but THC activates the CB1 receptor, and that's the key event that needs to happen in the brain for the classical effects of the cannabis high to manifest. And CBD does not activate the CB1 receptor. It is what is often called a negative allosteric modulator, which is a fancy term that means that CBD does not stick to this receptor and activate it. It sort of sticks to the side of the receptor and changes its shape so that it can no longer be activated as well by an agonist, an activator of that receptor. So in the case of THC, if it gets to the CB1 receptor and there's nothing there, it will stick to it and activate it. However, if THC gets to that same CB1 receptor, but CBD is already sticking to it, that receptor is no longer able to accommodate the THC. And so that THC will, effectively bounce off and either be degraded or maybe go float over to a different kind of receptor that it might have affinity for. And so there is a pretty good pharmacological explanation there for why there would be this interactive effect. They both interact with the CB1 receptor, but they do so in very different ways. And CBD can effectively get in the way of THC doing what it normally does at the CB1 receptor, which is going to mean it either gets degraded and simply does not do that, or it might essentially allow that THC to have time to go do something else at a different kind of receptor. And that's more or less what the best explanation that we have for why THC and CBD, when taken at the same time, is going to result in a different effect than THC alone, even if it's the same dose, right? So if it's 10 milligrams of THC versus 10 milligrams of THC and 20 milligrams of CBD, I think there's a reasonable chance that you'll notice a difference.
Awesome. When it comes to, your take on just kind of the commercial cannabis market, in terms of finding, products that have the, different like terpenes and different compounds that you want to be getting from cannabis, there's so many different ways nowadays, especially for people who live in states where cannabis is legal recreationally, as far as oils, topical, in edibles, the edible oils, flour. Like, what do you have an opinion when it comes to kind of the preferred means of getting cannabis, and whether you take like an isolate versus like a full spectrum extract?
Mm-hmm.
I would assume have very different effects.
Yeah. I would say, because there are so many different types of products out there, and because the naming and the labeling of these products is not as disciplined as you would like to make this as easy and reliable as it should be for consumers. No matter what I tell you or anyone else tells you, you're going to need to do some experimentation, and it's going to take some trial and error. Ultimately, what you need to do is find something that works for you, and then stick with that product from that brand. A product that's being reliably made and it's got the same effects and same formulation each time is going to be important. But I would start to think about things this way. First, you want to assess whether you want do you need cannabis because you want, is there something sort of wrong with the full body, or some sort of full body effect you want? Do you need the cannabinoids to get everywhere in your system, or is there something localized going on? So for example, if you've got knee pain and that's what you're looking to help, you plausibly will find help with something that you inhale that goes into your bloodstream, it goes everywhere. But you might be interested in something that you can apply locally to that knee. And so that's when you think about something like a topical, right? So a topical product is really appropriate when you don't want to feel psychoactive effects, especially intoxicating effects, and you've got a localized like pain or inflammation problem. So, and there's many different types of lotions and topicals that are out there, but that would be why you pick a topical. So, for example, I advise a company called Dosist, which is, as the name implies, very much concerned about precise formulations and controlled dosing. They've got some very interesting topicals that come in two basic forms. There's like an actual lotion, and then there's like a spray, almost like a, yeah, I mean, there's like a spray bottle. And one of them, the lotion would probably be best for, if you're at home and you really want to rub a lot of lotion and let it soak in and let it sit. And the spray is really more for if you were like on the go. So, variable one you want to think about is, do I want something that's localized or do I want something that's systemic? If you want it localized for pain and inflammation, that's, I would say, really like the best use case for a topical. So you can look into lotions versus sprays, and things like that. And you also, this is where it gets tricky, is just because something has THC or CBD or both in it, the the key is really, is your body actually going to end up seeing those things? And what's important in the topical space is a topical that has THC in it and or CBD is not necessarily going to have the formulation that allows those cannabinoids to penetrate the skin and sort of get where they need to go as effectively as they can. So a lot of them probably won't. Like if you just kind of put THC and CBD in, some, run of the mill oil for making a topical, a lot of that THC and CBD might not really kind of soak into your skin very well, and you effectively won't see it. What I can tell you is that the Dosis products have formulations that actually have other natural ingredients in there that help with the penetration through the skin. So they help the cannabinoids, which are fat soluble, actually sort of soak in to your muscles and kind of get where they need to go. So you want to be very mindful of the ingredients, even beyond the core active ingredients. So if you're shopping for a topical and you're not an expert in this stuff, you might buy two different topicals, see which one works, but pay very close attention in the one that works to what's on the ingredient list. There might be things in there that are allowing those cannabinoids to get in and work. But let's, the more common use case is, someone who wants to have a systemic effect. Maybe you've just got like full body aches and pains. Maybe you just want to feel good and just want to relax. One of my favorite pastimes is eating an edible about a half hour before I get a massage. I mean, everyone loves getting a massage, and I can only speak for myself, but if I take 10 milligrams of I like to take like a 1-to-1 or 2-to-1. So that would be like, actually, I think the gummies that I use might be 3-to-1. So it's 3 times the amount of CBD is THC. So, approximately 30 milligrams of CBD, 10 milligrams of THC. Like if I eat that on a relatively empty stomach about a half hour before a massage, it's a really good massage. So if you want that full body effect, you got to have something that gets into your bloodstream, and a topical won't do that. So you've either got to eat something, you've got to put something under your tongue, or you've got to inhale something, right? So an edible, tincture, or an inhalable. So that might mean vaporizing flower or a concentrate. And then, the way that you think about that is really just time course of effects, I would say. As many people know, but I'll say just in case they don't, the edibles tend to feel more potent. There's different reasons people give for why that might be. When you eat something, it's not going directly into your bloodstream. It's going to be metabolized by your liver before it gets into circulation. There's some reason to believe that THC, for example, can be processed into what is effectively a more potent version of THC. And that might be why edibles are often perceived as much more potent than inhaling THC. But edibles will be slow onset and slow offset. So that's really the key difference between an edible and an inhalable. It might take a while for that edible to hit you, depending on how full your stomach is and what you've eaten that day. It might be 20 minutes, it might be 2 hours. But it's going to last for several hours. So I know a lot of people who will take an edible dose of something before bed if they're looking to help with their sleep, because it's going to last all night basically, or at least several hours. An inhalable won't last as long, but it's also faster onset. So if you want to feel it, whatever you're looking for immediately, that's where you would look to an inhalable. I always recommend vaporizing more than smoking. Vaporization is going to be the healthier alternative. It's going to contain fewer nasty things. A lot of the nasty stuff in smoke is a byproduct of combustion itself. And it's not limited to cannabis, and it's not limited to tobacco. Anything that is burned is going to contain toxins. So, for better or for worse, if you like to go camping and sit around a campfire, you're going to be breathing in toxic wood smoke. So for that reason, vaporization is typically recommended, at least by me. It just means you're heating up the cannabis at a lower temperature than you would if you were applying a flame to it. And that actually allows all of those cannabinoids and terpenes to vaporize, but you're also not going to destroy them or transform them as much as if you touch an open flame to them. And of course, if you inhale something, the onset is more or less immediate. You're going to feel that right away. So if you're looking for that immediate feedback and you want that systemic full body effect, that's where you would think about an inhalable. And all I would say for flower versus concentrates is make sure you start low and go slow, especially if you're trying that concentrate for the first time, because it is concentrated. It's got more THC in it. I think it's a lot of it is just preference. How potent do you want it to be? How flavorful do you want it to be? The concentrates will be more potent. They can also be more flavorful because the terpenes are concentrated as well. The vapor might be a little bit more harsh. The more terpene content you have, the more it's going to be a little bit scratchy in your throat. In general, you might think about, do you want the isolated product with just THC, like a distillate product, or do you want a full spectrum product? I'm not in the business of telling people, which side of the holy war they should fall on. Some people are, diehard distillate fans, and some people say whole plant everything all the time. I think it's really up to you. You have to find what works for you, and a full spectrum product that contains THC and other cannabinoids and terpenes often will be better. It might have effects that pure THC doesn't, because you've got those interactions between those different cannabinoids and terpenes that may be there. Those will often be more flavorful too, right? A full spectrum product with terpenes in it is going to taste it's probably going to be more tasty than something that's just THC. That being said, that doesn't mean it's going to be the best product for you. Again, ultimately you want to think about all the things that I've just said, and then you really do have to figure it out with trial and error. Ultimately, this is a subjective thing. At least if for something with a psychoactive effect, and you need to try, and if it doesn't work, try something else, and if it does work, pay attention to what that product was and what was inside of it. So think about the dose and start low. Think about the modality. Do you want something systemic or local? And think about whether or not you want something that's long lasting with slower onset, or you want something that's fast acting and immediate.
Awesome. Well, Nick, we're coming up onto the end of the show. I've really enjoyed our discussion today. The last question that I have for you, you know, are there any specific areas kind of going forward in cannabis research that you're really excited about, either that are going to be studied or that you personally think really need to be studied?
Mm-hmm. Yeah, I think there's a lot of exciting things going on, such that, 5 years from now, we will know a lot more than we do today. So I'm excited for the paper that I mentioned that I've been working on to come out, which sort of just maps out the full chemical landscape of commercial cannabis and shows everyone what the actual cannabinoid terpene profiles are that are out there in the products that consumers are using. I think, it'll be really interesting to see how that informs things like the development of new chemovars of cannabis that people might breed, and informs studies in humans that are actually going to look for some of these interactive or so-called entourage effects. So, I'm really excited to see ideas like the entourage effect tested in both animals and in humans, and there's people doing that. So actually giving rodents or giving humans particular ratios of different cannabinoids and terpenes to test the idea that these different ratios can have discernibly and measurably different effects on people. So I'm really interested to map out that chemistry of all the products out there, and then map out what the different effects of these different chemical profiles actually is. And there are, like I said, there are studies going on with that. So in the next few years, we will have some insights. I'm also really interested to see, in the private sector, people develop new kinds of strains and products with brand new profiles that aren't really represented in the consumer landscape today. Strains that have much higher levels of some of these minor cannabinoids like THCV. There's some early preclinical hints in the literature that these compounds might have psychoactive effects, that they might have very interesting effects on metabolism and other physiological effects. And it'll be really interesting to see products actually get developed and put out into the world that actually have these sort of brand new profiles. So, I'm generally interested in the entire area of just the chemistry of cannabis, mapping out what all of the chemical phenotypes are, and then really, seeing people test what the effects of these things are in systematic ways, right? Like, right now we have a lot of folk knowledge in the cannabis industry. There's a lot of belief that's based on experience, that's also highly subject to expectancy effects and our biases about these different effects that these different types of cannabis cause. And I think 5 years from now, we'll know more. We won't know everything, but we'll know more about whether there's really something to these claims, and more importantly, like what the actual ratios are that cause these different effects. So that's sort of what I'm keeping an eye out for.
Very cool. Well, Nick, if people want to, get in touch with you or find out more about your work, where would you direct them to?
The best place is my website, nickjikomes.com. So that's NICKJIKOMES. That is my personal website. I occasionally put some of my thoughts in writing there, but it's mainly a place where you can find all of the audio and video episodes of my podcast. So I have a science podcast that I put out each week. It's about, basically anything science related, but I have a bias for talking about psychoactives and drugs. I have a bias for talking about, emerging types of biotechnology, and a lot about like neuroscience and the mind. So that's where you can find me, nickjikomes.com. You can also on that website find my social media. I'm relatively active on Twitter, posting things. And if you want to sort of learn more about what I'm thinking about, the podcast is probably the best thing to check out.
Awesome. Well, if you guys enjoyed the show, go ahead and like and subscribe to our YouTube channel. We're Roscoe's Wetsuit, and you can also find audio versions of the podcast available on Spotify, Apple Podcasts, Stitcher, and just about anywhere else that audio podcasts are available. If you guys did enjoy the show, I'd really appreciate it if you could leave us a 5-star Apple Podcast review. That would be really helpful. Nick, again, I wanted to really thank you for coming on and sharing all your expertise and knowledge, and I really appreciate your time today.
Thanks, Toby.